He was supposed to speak in the next session, but you have to bring him forward. That would be Michael Landy, Kofi Landy. Michael, if I'm just gonna make you the co-host and please unmute yourself. Michael is at the International Institute of Tropical Agriculture in Kenya and is also a PhD student at SLU, Sweden. He'll be talking about the genome assembly of the white star apple as part of the African BioGenome Project. So, Michael, do you want to unmute yourself, please?
Yes. Can you hear me okay?
Yes, please, we can hear you.
Perfect. Hi, everyone. My name is Michael. Just allow me to put off the video just in case I experience any internet connectivity. So we'll start by sharing my screen. Okay. Mm. Can you see my screen now?
Yes, please, we can see your screen.
The whole, okay, perfect. So let me
Yes,
just
carry
get
on. Yeah, carry on.
All right. So as thank God mentioned, I'm based in Nairobi, Kenya. And today I'll talk about the African star apple genome. But for this talk, I will not talk about the introduction of this crop. I will dive into the genomics. We will have, I think, tomorrow 2 talks on library preparation and also propagation of this plant, this fruit. But as you can see, it looks a really delicious plant, and I'm really looking forward to visit Nigeria to test this fruit, the star apple fruit. When we think about plant genomes, we think about stories that we either talk to our children, and when you read a story from a book, that will be probably the genome, and the pages would be sort of the chromosomes, and the pages that you read will have the instructions that probably have you think in the genome world will be the genes that sort of give us the instruction, the functionalities of of uh, of our plant, and why we care about plant genomes is these are just examples of the applications of plant genomes, and just to mention a few, if probably you want to think about crop improvement and breeding, sort of looking at wild type plants and maybe transforming them to domesticated plants. Obviously, plant genomes come in handy if you're thinking about resource conservation and ecological resources. I think this is probably the main theme of this workshop. So genomes come really in handy. If you're thinking about plant evolution and classification, genomes also are really, really important. And just to mention about epigenetics, thinking about beyond genetics, then you have to also understand the genome to explain any traits that are influenced by epigenetics, or rather the environment. So in the world of genome assembly, I think probably Tassian already mentioned, but think of it as solving a puzzle where you have little pieces that you try to join together to have a bigger picture, and that is the genome. And just to give an overview of genome assembly is these small pieces in a jigsaw puzzle would be your short or long reads So small pieces of DNA, I'll explain how they're produced in the coming slides. And as you're solving this puzzle, you tend to join these pieces, and these reeds, you join them to form contigs, sort of bigger, smaller chunks of these small pieces. And these contigs you can also assemble them to scaffolds, and later on you have a full representation, for example, a chromosome, and within a genome, say, for example, with the star apple genome, you'd have all the chromosome, and then you can define actually the functionality of a plant from a plant genome. But while we always do this the process of genome assembly, there are always some problems that we encounter while assembling these plant genomes. Um, on this plot here, what you can see is, if you imagine this a plant is a diploid inbreed species, where you expect two copies of the genomes coming from the two parents are homozygous, then it's always easier to represent this genome as single haplotype reference assembly. And by haplotype, I mean one copy of a diploid. So a diploid will have two copies. So one copy will be the haplotype 1 and haplotype 2. And for similar copies, sort of homozygous genomes, you only need to have a single haplotype reference. And this is pretty straightforward. But when we have cases where we have outbred assembly, then it becomes a bit tricky. And you have, so here in gold and blue, these are contigs, or sort of a chromosome coming from one parent, so the maternal and the paternal. And always when you assembling these pieces, you sort of don't know where the pieces are coming, either from the father or the mother. And you can always represent this either as a collapse representation, and how we call this we call it as haplotype mosaic reference, where you represent this as combined if you cannot really differentiate which contig is coming from which parent. But in the recent times, we do have some well-refined sequencing platform that I'll mention in the coming slides, where you can really define where contigs or scaffolds come from. And then you get a haplotype first reference assembly, where you know that, okay, this haplotype 1 is coming from this parent, and this other one is coming from the other parent. And this is specific for diploid, an example of diploid genomes. And so, as I mentioned, the long reads sequences, and here I specifically mentioned the high 5 reads that are making us plant genome assembly to be really easy. And the fact that, and I will not get to the details of how this process goes on. I think we'd have a presentation on library preparation and get to understand it more. But just to point out here, the hi-fi reads have very high accuracy, an accuracy of more than 99% accuracy. And with this accuracy, you have the advantage of accuracy and also having long reads. And if you have these two combination, then it's really easy now to assemble a genome and to have really high quality genome assembly. So just to jump in on the workflow of the genome assembly of this species star apple, and just to mention here in green, these are the steps that I will share. So once we have this hi-fi reads, we do the quality control, because this organism has not yet been sequenced and assembled. It is always nice to estimate the size of the genome and also know the ploidy level, how many copies of this genome in these plants. And I will share some of the assembly statistics after doing assembly using this tool called HiFi-SM that I will share the different steps. And then check for duplications and remove if there's any duplication rates within the genome scaffolding. And in the end, in red here, these are steps that we are ongoing. And I need to mention that this work is ongoing. So some of the results are preliminary results that I will share. And so after scaffolding, normally what you want to do is you want to do manual curation. You want to gap fill and remove any contaminations that are present in the genome. Evaluate the assembly, and then obviously at the end you want to look at the gene repeat and the repeat annotations within the genome so that you can explain some of the functionalities of this plan. And so the data that we use in this case is the PacBio HiFi reads that I mentioned are really good quality. And we sort of use additional information that is the OmniC. These are the um, reads or data that we use to do the scaffolding part of the analysis. As I mentioned, the quality is really good. And here I'm showing, so the first plot is HiFi reads, and in the bottom is the OmniC reads. And here is just the length of distribution of your library. And what is interesting to note, we always have reads that are way too short and reads that are way too high. And the first thing what we need to do is to filter out those extreme lengths. And what we did is we filtered the HiFi reads based on the length of 10 kilobase pairs and to 30 kilobase pairs. And for the OmniC reads, because these are short reads, and so we only filtered any read that is 50 base pairs and above, and that's what we kept. And here I don't show, but we filtered out some bases upstream, so 5' upstream, around 15 base pairs. And for the OmniC also, we trimmed 14 base pairs upstream that were a lot of biases. And what I mean by biases is we couldn't, from the quality control, you can always see unproportionality of the bases within your library. And so we ended up with a total number of reads for the HiFi reads, 3 million, and the length, as I mentioned, 10 to 30 KB. And then this was the also total length of the reads for the OmniC that we take to the downstream analysis. Now, for the first step is to, once we have our data, our clean data, what we want to try to answer is what then is the estimated genome size and what is the ploidy prediction. So what you see here is a plot from a tool called GenomeScope. And here, what you're seeing, these are KMA plotted. So this approach will try to estimate the size, and here it tells you the estimated size of this genome in one copy is around 822 megabase pairs. And you could already see that is a very highly homozygous genome of 99.7% and very low heterozygous level of 0.3%. And already here you see like the first peak will be your heterozygous KMAs that are pretty low from the percentage. And the second peak are homozygous KMAs that shows this genome is highly homozygous. And we used other tools called SmartPlot to just predict the ploidy prediction. And as you can see, so we use different types of KMA sizes to do the prediction. And as you can see towards the end here, AB, it estimates 82% that this genome is a diploid genome. And if we also do another trial with 31 kmer, then we get 78%, which sort of these two approaches tend to confirm that this is a diploid species, and the estimated haploid size is 822 megabase pairs, and it's really a highly homozygous genome. So after this prediction, we went ahead and do the genome assembly, and here I'm just showing the method HiFi-SM. We used additional two other methods to assemble these reads, and I will share the statistics, but just to focus on HiFi-SM because this was the main output that we take for the assembly. And what happens here is you have your input HiFi reads, and as I mentioned, you'd have a set of reads coming from here in orange is from one haplotype, and then here in blue is from the other haplotype. And so this region here is an example of an heterozygous variant. And in the green, these are homozygous reads, so reads that are present in both haplotypes. And so what the tool does is first it will check for any sequence error and correct this, and will identify sites that are actually the heterozygous alleles. As you can see here, there's AAA, and from the other haplotype is CCC, which is the different allele. And so after it corrects, it sort of starts to build a graph of this genome. And these bubbles here are where we define reads coming from different haplotypes. And so once you get to these graphs, you can either go two way. Either you get the primary assembly construction where you only have the primary with one of the variant and have sort of some alternative assembly, or if you have other complementary data, and these are actually reads coming from both parents that help you to separate these two faces, which we didn't have for our case. And so we ended up representing this genome as a primary representation. And also the fact that it's highly homozygous, then getting the primary assembly of the genome was an ideal approach. And so after doing the assembly, we now did some statistics, and what you can see is a screenshot of the statistics of the genome. So the first row is outputs, the primary assembly of HiFi-SM, and HiCanu, which is another tool that we use to assemble this genome, and file is another tool that we use. But just to draw attention on three key characteristics. So first is the number of contigs that the assembly generates. So you can already see around 7,111 contigs are only for the HiFi reads. So what you'd expect is you want a lesser number of contigs. So the HiCanu and Fly tend to have a lot more contigs of the assembly. And in terms of the total length, well, this was really surprising because we already saw the estimated genome was around 820 megabase pairs. But here we are seeing a huge number of total length. So this is 2.4 gigabytes. Even more for HiCanu and Fly. Fly got 15 gigabytes, and this was 2.8 gigabytes, which is really, really huge. And also another factor to check is the N50 value, which was really good for HiFi-SM, 1.3 megabase pair. So this tells you the contiguity of the assembly. And for the HiCanu and Fly had very low, and this sort of already showed that these two assemblies here were highly fragmented. And that's why we move forward with HiFi SM assembly. So then with this primary assembly, we went ahead and checked for the completeness of the genome. And tools like Busco will tell you how complete this genome is. And you can see here it's 98% complete, but then the duplication rate is really high, 87.6%. So this was really puzzling for this genome. So what we do next is we look for regions that are highly duplicated. And what we use is we use a tool called purge tubes that has two main steps, where the first part, what it does is it takes your long reads and then maps it to the reference that did primary assembly. And what you get is sort of a coverage output. And then the next thing, what it does, it splits the assembly into chunks of say 10,000 base pairs, for example. So split assembly by a certain number of length, and then it sort of does a self-alignment, right? So using the self-alignment and the coverage results, it checks for haplotypes, or rather contigs that are identical, and filters this as exact matches. And so it removes this duplicate this way, and you'll end up having a purged genome, and some of the contigs that were actually removed. And what is interesting with this tool is you can redo these three steps until you get a decent haplotype set, haplotype set that is either less duplicated, and it's also close to your estimated value. So we did this step, and then we moved forward and now get the statistics of the draft assembly. And here is the same that I mentioned before. So we have the number of contigs. So the first column is the primary assembly, and then the first round of purging, so the purge gap, and then the second round of purging. And you can already see in the total length from 2.4 gigabytes, then we get to a close estimate of the genome, which is around 821. And in terms of the N50 value, the last column, as you can see, is 3.1 million, which is really improved contiguity as compared to what we had in the draft genome. If you look at the duplication levels, so for the second round of purging, we have a 6.6% duplication rate, which is actually more expected if you compare with even other genomes that have already been assembled. So this is really good. So we move the next step, where we do the scaffolding using now the OmniC reads. And for the statistics again, for the star upward scaffolds, we now have 596 number of scaffolds. The length is 821 megabase pairs, which is really what is close to the expected size. The N50 is 57 million, which is highly contiguous, and this was good. And already with the L90 value as 13, this sort of gives you an idea of the number of chromosomes that you'd expect for this species. And normally it's always +1 or -2, +1 or -1. And just here to show is a contact map showing the scaffolding of this genome. And the next step, what we do is then to do the manual curation of this genome. So once we've done the scaffold, so we need to manually curate this genome. And then because we have the scaffold, so we expect to have gaps that we need to fill and check for contamination within the genome. And we will submit this genome to NCBI, and later on do the genome gene annotation and just to have an idea of the repeat content of this genome. And a point that I didn't mention is having the write-up and submitting it to be accessible to everybody. And just to conclude, so we expect to achieve a very high quality of star apple genome, and this is the best, the fact that we already have high quality reads both on the OmniC and the hi-fi reads, and this would actually be relevant in the genome-assisted cultivar breeding to provide a better understanding of important metabolic pathway. And we will actually see some of the benefits of this crop in the coming days from the other presentations. And we would also use this data to do some capacity development where we will do some training, workshop on genome assembly and annotation to build regional capacity and promote the intercontinental transfer of knowledge. And now I'd like to acknowledge everybody who's involved in this project, the INCABA who did the sequencing, and we will hear about them in the coming session. African BioGenome Project, led by Thank God, for actually organizing the whole workshop and also managing the whole analysis. IITA, where I'm based with my supervisor Andreas, and the team from ANCAB and IITA who have really assisted in making this a success. So thank you very much, and I'm happy to take any question.
Well, thank you, Michael. This is this is very detailed and good to see the progress. So we're just gonna, if you could please stop sharing your screen, Michael, so we can have
Yes.
the two screens together. We're gonna take questions. So do we know if people on site, do we still have people on site? Control? Is Control still on site? Are they still online, or just trying to see? Oh, it looks like we lost the on-site people, the connection. So we're going to take questions from those on Zoom, the participants on Zoom rather. If you do have any question, please use the icon to raise your hand or put that in the chat box, please. So we have some questions here. So Hawan, do you want to ask your question orally to Michael, please? Hawan, let me try and find you. Yeah, Ewan, you can unmute now and speak, please.
Okay, thank you very much. I was watching and listening and learning a lot, specifically this star genome plant is really has a reference genome. It's because this is my childhood fruit, which I only know eastern part of Ethiopia. And this is really great resilience, and the wood is really great. But I didn't find that no research at all. And recently we are collecting this germplasm to disseminate to the northern part of Ethiopia, because as you can see, in the northern part there is a lot of degradation, and the forest is shrinking. But this is a fruit and by itself, and the wood is really great. And I'm so much happy to find out, lastly, the Africans are really sequencing this genome. So my question is, when you are doing the reference genome, as you know, the distribution of this plant is in the equator, I think around that, and a little bit to the north and a little bit to the southern part of Ethiopia. No, to the southern part of Africa. So when you are doing the reference genome, which accession or a variety have you used? And is it like, a kind of, can we trace the plant passport data? That will be really interesting. Then it will help us to proceed with our research in this plant too. Thank you very much.
Okay, that's really nice to know that the fruit is you grew up taking the fruit. That's really nice. So onto your question, I'm actually really looking forward to actually test the fruit itself. Yeah, so to your question, so I think the accession was from Nigeria, but I think we will get more of the presentation on propagation of this plant tomorrow. Yeah, so maybe then we can have I think the accurate accession of this plant. I don't know if thank God you know, because I have no idea.
Yeah. So, Hewan, this is from the IITA collection, the accession, and the presentation on the wet lab and sample collection and processing part
Sample.
of this work
Yes.
will be presented tomorrow by the IITA scientists and by the genome facility where it was done. So you will get more background information on that. But definitely, it's the collection at the IITA. It's been there for a while, and then yeah.
Thank you.
Okay. Yeah, it's
Yeah.
good to see that you like the fruit as well, right? I'm sure many people on the call. So for many people who perhaps cannot map the common name to the English name, also this is agwalumo or udara, for instance. You know, for many people who know the local names of this species, so it's hawked everywhere on the streets in West Africa. And as you just rightly also pointed out, Hawaii is also found in Ethiopia, which is good. Okay. Thank you so much, Michael. We have an additional question. Professor Anemugai has a question, and you want to go ahead, please?
Yeah, I think for me, I just want to appreciate the two scientists because it's showcasing the fact that we can do this work on the continent. My question to both, very briefly, you've been able to present very high quality data. It'll be interesting to hear about the start and whether it was as easy as you have projected it to be. What are the challenges, for instance? Just off the cuff, what are some of the challenges you faced? Was it the sampling? Was it the isolation of the nucleic acid, or what specific challenge did you have? Thank you.
Maybe we can also spotlight Ansia here. So it looks like this is a question for both of them. So Ansia, if you want to put on your video or something, so I can spotlight you if your video is off. So I can do it and then you can go take it back as well. So you want to put on your video, Ansia? So that I can spotlight you and yeah, okay, found you. All right. So this is more of a question. There's an important question, Professor Mugai, on what it is. So I'm sure you both heard about it. So you want to just talk about it, and I'm sure that would also help inspire others. Because one of the things Africa BP aims to do is to sequence the genome locally. No matter the challenges, no matter anything, it has to be local. And of course, many people already have seen that. So let us know in the next 5-10 minutes. I want us to be very detailed, explanatory, and yeah, just say over to you.
And so do you wanna start or I can?
Sure, okay.
Yeah.
So some of the challenges we face, so I am not a plant bioinformatician. I work mostly with humans and animals and pathogens, so it was very new for me to work on plants, but Africa BP has been really great in, linking me with all the right people because you don't know what you don't know until you realize that there's more to know. And another challenge that we specifically faced was computing resources. So because the plant that I was working with was so complex, a lot of the typical resources that we throw at assemblies wasn't enough to resolve this complex genome. So I'd had to go, just speak to other African researchers and to see how they do things and how I could improve how I was doing it. And yeah, we've made some good connections with thank God's team as well. I think Sadiq has been very helpful, and I've learned a lot through the whole process. I'm just very grateful and happy that we can sequence African plants on African soil and analyze it ourselves. Over to you, Michael.
Yeah, I think I would say the same. the complexity of the genome. For my case, I've worked on cassava. For some reason, it was a bit straightforward having the reference of cassava. But this one also, like it's a new genome that has not been sequenced before, so there is not much reference to compare. And even like you go high on the family level, is then we get species that have been sequenced. So there was not much to like compare. So you actually starting from scratch, and the complexity of the level of duplication that was really uh, sort of challenging. But of course, with the network, Sadiq also like really helped in the discussion. Thank God to within the African Assembly group. So this was really helpful. And also just to mention, like I'm really completing. I mean, the last year of my PhD, so a lot going on as well. So then this was really I think a lot of work, and hopefully now we're getting to the end of it. So this is really nice. And actually sequencing, I really like the idea, thank God, like sequencing in Africa, doing it in Africa, collaborating within Africa, and building capacity within Africa. So that's really nice. Yeah.
Yeah, thank you, Michael and Ansia. I think this has also been very interesting because it allows us to see firsthand what the challenges are, and also allows us to see and experience firsthand how we can actually drive and move things. I mean, looking at what you guys have done in this genome, I was telling someone the other day, they said, "Oh, thank God you called me in." I said, "No, I think the genome is now on autopilot in the sense that we now need to publish it. So everything, you know, so it's actually been very fantastic. And what awaits you guys now is how do you now take those bits, and then after it's published, take those bits and also um, uh, uh, increase it in a geometric fashion in terms of knowledge, in terms of giving to people, in terms of, making it open, giving people the opportunities that, the experiences and sharing it, especially on the African continent, um, because one could sequence all this. There are institutions outside Africa that can sequence all this in a year or 2 years, but we said no, we don't want that. We want to sequence it, and I've seen people who have also built their opportunities by just doing it because you have that sense of ownership, confidence, and things like that. So I'm really pleased with this. So there are also many of those opportunities and projects within the Africa BP also in that same direction. And most of the time, I think to myself, what if we can scale this beyond just the number we have? What if we can take it times 10, times 20? Then that would then get us closer to the Africa we want, which all of us on this call, I believe, do want. And also that will also increase the number of researchers, for instance, because Africa has the lowest number of researchers globally. An example is that the average number of researchers per million people is around 1,400 researchers per million globally. But Africa has just around 300, 350 per million people. So which means the number of people that can actually drive things low. So we need to then increase it. And I think that is one of the significance of a project like the African BioGenome Project. And I really want to thank you, Ansia and Michael, for also showing that this can be done. And I hope that this will also provide a sense of motivation and inspiration for everyone on this call. This is a high quality assembly, high quality activities, and lots of things that experiences shared over the past few months. All right,
I think I want to just say that I think we shouldn't give up because from what we have seen is that the project started a bit slow. Ansi, I think you also experienced that slow start as with Michael, a lot of retries, and thank you for not giving up because eventually it worked. And that's now we're looking forward to, implementing that scale globally. Yeah, thank you.
Yeah. And I think you guys didn't give up because at some point you were saying, "Oh, like, okay, all right, that's it, right?" So, what especially with all of this deploy polyploid, it's just seeing the next species and doing something with it. That is where the whole thing is. All right, thank you, Anne. Professor Anne, this was supposed to be a comment, but then it's turned into a panel session. And all right, thank you so much, everyone. Michael and Nancy, I hope you still hang around. We have a few more, 1 or 2 more presenters, and then we can wrap up for today. Do we have, actually, we have people on site. Do we still have control on site? Professor Julian, do we still have control on site?